Thank you so much for that incredibly generous introduction. I've so missed being here virtually, not with you in person. I have so many good friends at UK, and Linda's right up there among all of them. And what I want to do today is take away that question mark at the end of this. I want you to hopefully share my enthusiasm and optimism at the end of this discussion this morning, why I'm so excited about the future for Alzheimer's disease. It's been quite a journey, and I've spent my entire career, in this area. And it's such an honor to, be participating in this Markesbery Symposium. The University of Kentucky ADRC, the Sanders Brown Aging Center, just played such a central role. And I would say Bill Markesbery was ahead of his time. And Linda and the whole team there continue to contribute so much progress nationally that have inspired me personally. And so it's, again, thank you for the opportunity. Let's see if I can get my slides to advance. I'll be in good shape. There we go. Let me just first mention a variety of disclosures, because I'm going to be mentioning just a few things. I want to give an overview, very high level overview, and touch on some of the exciting things that I see. Among the things that I'll mention will be, I'm going to have to set the stage for what Greg is going to talk about in the panel with the new medicine. Can't talk about Alzheimer's disease without at least introducing that. And I'll be mentioning also an advisor for a company called Cognito, which is a novel way to do treatment. So I'm just going to be mentioning some of these. And what I'd like to talk about, frame things, is, while I know you're all here because of your interest in Alzheimer's disease and related conditions, I'll just make sure that we're all sort of starting at the same place, including where we are with current treatments. Linda mentioned the Accelerating Medicines Partnership, which has been a major source of our inspiration and effort over the last 5 to 7 years. And it's important to me because it's helping us think about how to make sure that we can detect the disease as early as possible in the future, and then think about how we can expand the treatment pipeline. And so I'm just going to again touch on things that I'm sure are ongoing everywhere, as well as highlight some of the things that we're doing at our center, which complement in many ways all the exciting things happening at University of Kentucky. So it was over 100 years ago now that the first case, so to speak, of Alzheimer's disease was discovered, and it was given its name because Alzheimer's saw under the microscope these two hallmark pathological changes, these amyloid plaques and the neurofibrillary tangles, which are now pathologically how we how we confirm the presence of Alzheimer's disease pathology. And these have been, so while they've been recognized for over 100 years, so much of the field has focused on these two pathologies. And by doing that, our field has learned a lot about the natural history of Alzheimer's disease. And unfortunately, I know many of you know too well. This is a journey. We have spent a lot of time trying to recognize the first clinically evident time when symptoms began, which we term mild cognitive impairment. And for people, it's a journey typically that begins with progressive memory loss, expanding into other cognitive areas that become impacted and affect functioning. And too many of us think about this later stage of dementia when people really need a significant amount of help, assistance to live, to feed, to take care of themselves. But this is a chronic disease. I think is one of the things that's really changed the way we think about the disease. And even by the time people begin to present to our clinics with mild cognitive impairment, there's considerable injury in the brain. So there's already been shrinkage that's often evident even by the time people become symptomatic. But most importantly, I think all of us are now very excited about this large window of opportunity with the realization that these pathologies that Alzheimer's described, we now know, as we'll talk about, these begin 20 or more years before the very first signs of memory loss that really become apparent. So it's, from one hand, it's a little bit frightening that changes. There are 50 million people in the United States walking around with pathology, it's estimated, in their brain of Alzheimer's disease, even though they're completely normal. But you know, the important thing is that there's this long window of opportunity before symptoms begin. And that's why so much of the work that's ongoing now, I want to try to give a broad perspective on, I think, lots of opportunities to think about how to do things better. We often call this a silver tsunami because Alzheimer's disease really affects almost 50% of people after they get to the age of 85 or so. And because all of us are living longer, the population of 85-year-olds is growing rapidly. You know, we're going to see doubling and tripling of the number of people with Alzheimer's disease and related conditions. And so this is a huge toll it's taking on society. But of course, the greatest toll we know is that to the individuals and the families that are affected by the trajectory. And it's getting worse, so we've got to change this. You know, what's happening is we've seen the impact of important research across other areas of biomedicine, cancer, heart disease, and stroke. And as a result, fewer people are succumbing to death as a result of these other diseases. We've changed cancer from a disease that was just, you know, everybody feared with no treatments to now a highly treatable condition for most people. But what's happening is people are living longer, surviving other conditions. Public health has improved, and Alzheimer's disease continues to increase. So we all know we're in a situation which inspires us to do better. We've got a lot of people affected by the disease. The costs are terrible, individually in society, and the treatments we currently have are just inadequate. And you know, with all this background, our country as well as others have begun developing national plans. And so we're at the 10th year anniversary now of the national plan in the United States that was initiated when Obama signed it into law. And the goal, the first goal of that is to prevent and effectively treat Alzheimer's disease by 2025. So already 10 years into this journey, and we have a lot of work to do if we're going to achieve this goal of preventing and effectively treating Alzheimer's disease. The Sanders Brown Center, our center, and others have been really fortunate because the increased awareness of the problem of Alzheimer's disease has led to this major growth of research dollars available. And so here in 2021, we have now over $3 billion a year going into research in this area, which is still not enough, but it continues to go up and is expected to go up because of the bipartisan support. And every couple of years, there's a convening of scientists and clinicians from around to help establish the priorities for research, to make sure that under-researched areas are invested in that have great promise. And all of us in the field have been beneficiaries of this infusion of dollars and the research opportunities that are prioritized. This is just to remind everybody that we have a lot we can do right now. When we do early diagnosis, I've always been a strong believer that that's absolutely essential. It pains me that we don't have better screening in the elderly for cognitive impairment because there's so much that can be done. Establishing a correct diagnosis to rule out other conditions, and when Alzheimer's disease and related conditions are ruled in, we have treatments which are, you know, not as effective as we want, but they've been around for nearly 20 years or more, including the cholinesterase inhibitors that are known by names like Aricept, et cetera. And Namenda is another class of drugs which is available. And Alzheimer's disease is, I use that term Alzheimer's disease to really reflect a broad spectrum of symptoms and pathologies that occur commonly with the memory and cognitive changes. So we know depression and sometimes delusions and hallucinations can appear as part of the disease. And I think we also don't fully utilize non-pharmacological strategies that could be helpful in many cases to help people develop tools to empower themselves to improve their memory or keep it stable and to adapt as their memory or cognition changes. And we know many of you out there are in the role of caregivers and there's a lot of research that needs to continue in this front as well. We learn more about the needs that all of you have and how we can best meet them and address gaps. I always like to convey a lot of hope and encouragement. I think this is, again, a chronic illness, and there's so many years that people can do really well while we look hard and fast for better treatments that can hopefully prevent things in the future. So I like to end this treatment part by saying, we really haven't had any disease-modifying treatments that are approved. Why is that? And as I mentioned, we've known about these amyloid plaques and neurofibrillary tangles for 100 years, but the world has been focused on one idea, which has been an important idea, and it's because of the recognition that amyloid plaques are composed of this protein called amyloid, or A-beta peptide, and it's led to this whole theory that has galvanized the field and industry to think about how we can get rid of the amyloid plaques. And I think everybody has woken up after the last 20 years of clinical trials, which is a list of some of them, which I don't expect you to read here. But every single one of a multi-pronged approach to treat these amyloid, to prevent its production, to help clear it away out of the brain, and other strategies have really all failed. And that is what inspired a lot of the strategic plan nationally, that we've got to broaden the search and understand the complexity of what's happening in the disease. And just a couple of years ago, we started seeing really concerning signs that even the major pharmaceutical companies, who we absolutely depend on to help bring drugs to the clinic, started getting concerned. Because of the failure of the clinical trials and the large cost that goes into these clinical trials, people really started getting concerned. There was even alarm went up when Pfizer, one of the major drug companies, halted their research program in Alzheimer's and Parkinson's disease, and others stopped major programs. And so our whole field changed quite a bit last year in a very surprising FDA decision to consider approving this drug that Linda mentioned at the beginning, called aduhelm, or aducanumab. And it's a monoclonal antibody to the amyloid, much like many of those failed treatments. But this study, you're going to talk a lot about with Dr. Jicha in the panel later. And I just want to set the stage for that panel, because again, it's so important. The reason people have been interested in this drug, there was a phase 2 study, a small study, which was not meant to determine whether the drug would be approved or not. But it illustrates nicely what this whole class of drugs now is pretty well established to do. And what you're looking at here on the left are brain scans from 4 individuals in the phase 2 study, and you're looking at the yellow and red, which is the amyloid signal. It's where the amyloid plaques are developing. And you can see at baseline, when these individuals started, they all had lots of the amyloid present in their brain scans. And after 1 year of treatment with this drug in this study, those who received the placebo or didn't receive the active medicine had no change or worsening on the amount of amyloid that was accumulating, but in a dose-dependent way, the amyloid was cleared. And they also showed in this phase 2 study some promising results for the first time, that this change in amyloid was actually showing some relationship to the progression of dementia in a dose-dependent way. So that was very encouraging and led to the trials that Dr. Jicha is going to talk about that have become very, very controversial. And again, I have been a consultant for Biogen, so I just want to make sure that's disclosed. They probably don't like that I'm a consultant because I haven't been terribly enthusiastic. But basically, this was what they did was 2 large clinical trials where over 3,000 participants from many of the Alzheimer's disease research centers and other places around the globe participated, where they received this monoclonal antibody to amyloid for about a 1.5 year period. And one study was called Gage, and one study was called Emerge. And what was shown was what we have seen across many of these anti-amyloid trials. This is that the amyloid can be cleared. And I'm not going to go through the details of this, but remember there were 2 trials, and one of them showed what you're looking at here. I think is important to look at. And so the purple line going across here, those people who received the high dose of the monoclonal antibody, and the placebo here is in gray down here. And what we're looking at is over time, the mean change in the memory or cognition on a scale called the mini mental status exam. And over time, people decline, as we know happens with Alzheimer's disease. And there's this very small difference between the placebo and the treatment, which is what has been considered the benefit. So it's a very small change, and people don't often recognize that continually the disease is still getting worse. So it's not reversing anything at all. In this one trial, there was also some small benefit seen in how people were doing functionally. They did not lose functional abilities quite as fast as those in the placebo. But the almost identical study showed 0 difference between the treatment and the placebo. So that's why there's been huge controversy, which will be discussed in the panel, because usually the FDA requires 2 trials like these to both show significant benefits. And so again, that's where we are. And as Linda said, the FDA has approved this as a sort of conditional or fast track pathway where more data needs to be accumulated. So I consider that we still do not have any disease modifying treatments at this point that are clearly effective. Our own practice at Emory, we're not even prescribing this yet for patients. We may or may not do that. If we do so, it's going to be under only in very, very restricted conditions for people that would otherwise have qualified for this clinical trial. So you're going to talk a lot more about that. So that's pretty much all I want to talk about amyloid, because the exciting part, I think, is thinking about this going forward. But why are these trials all failing for the most part? And one of the major reasons is because the symptoms are occurring, as I showed you earlier, after a long 20+ year period when the pathology is there. And so we're starting these clinical trials very late in the process. Many of us think there are other targets that are important to go after other than amyloid, and we'll talk about that. And we still have a heck of a lot to learn about really what's causing the disease, particularly in these early stages of disease. And so part of this strategy then was to expand the pipeline of novel therapeutic approaches. And one of the initiatives that has been funded is called the Accelerating Medicine Partnership. And it's a partnership between several academic groups, as well as industry and NIH, to work together in an open science manner to produce data that anybody can access to help us learn about what's going on in the disease. And this program was inspired by thinking about the really exciting progress that's happened over the last 10 years in understanding the causal mechanisms involved in Alzheimer's disease. And we know this is a complex disease, that it is a combination of multiple genetic influences, the environment we live in, and unbiased approaches to try to find out what's going on have used genetics. So thousands of people are necessary to participate in genetic studies so we can understand which genes confer risk. This is even a short list now. There's nearly 40 different genes, which we all carry, that have different flavors. And depending on the flavors of those 40 genes that we have, our risk for Alzheimer's disease is either increased or decreased. So this is something we're born with. And epidemiologic studies also provide really a much more of an unbiased approach by studying large numbers of people and understanding what factors lead to people having an association with increased or decreased risk. And a whole host of things have come out repeatedly from different epidemiologic studies and different cohorts across different parts of the world. We know that age, by far, the older we get, the greater our risk. We know there are influences of gender and many other factors. And in green here, I'm highlighting that there's a lot of evidence that continues to accumulate that education, exercise, anti-inflammatory diets like Mediterranean diet or the MIND diet, are all things that can influence risk in a population base. So this is all what we consider association. So it's not necessarily cause and effect, but they're important clues. And if you look at this holistically, we've been focusing as a world on these amyloid as a target. The genetics has only pointed towards amyloid in a very, very small number of people with an extremely aggressive form of young onset Alzheimer's disease, and much less in the more common forms. But if we look at what these genes and the epidemiologic studies are suggesting to us, that many of these genes and risk factors seem to be associated with lipid biology, vascular biology. Dr. Estes and others at Kentucky have done great work helping us understand how these genes confer risk of the disease. And then there are also many genes and factors in these studies that suggest that inflammation is another one. So again, this inspired the Accelerating Medicine Partnership Program that we need to actually start with human brains. And there's nothing more informative than studying a person during life. So many of generous participants in the Alzheimer's disease research centers donate their brains at death, as well as controls. So people who have had different diseases as well as who are just healthy. That has become one of the most important sources of information for us to learn what's really going on in the brain. That we can then turn that information into better ideas to test other things in basic research studies. So we've now, as a consortium of different centers that are part of this Accelerated Medicine Partnership Program, we have actually analyzed over 2,500 postmortem human brains, where there's a wealth of clinical information available, detailed pathology that's done consistently, genetic information, and all these other molecular fancy ways to understand at a very precise level what the changes are occurring in the brain. Our team at Emory has been leading this consortium for proteomics, and I'll try to explain what that is very briefly. This is a cartoon showing how every cell in our body has a complete replication of the 20 to 25,000 genes that make us human. Every single cell in our body has that. And we're born with that blueprint of life. And each of those cells changes that genetic information into a product, and the product is a protein. And while there are 20 to 25,000 genes, these confer the instructions for cells to make over 1 million different forms of proteins. So it's very, very complex. But it's these proteins that are the ones that are the substance, the architecture of the brain cells, the immune system, and the amyloid plaques themselves are a protein. And the inflammation that occurs around it, and many are protein mediators of these things. And so proteins really in many ways are one of the final common steps in the execution or pathway, as I like to say. And so these proteins reflect the integration of the genetic and environmental influences that we just talked about. This is not going to be a scientific talk, so I'm not going to get into all the details. But this approach, allowing us to study for the first time at scale, we can measure about 10 to 12,000 proteins now in these brains, and thousands of brains, and begin to understand what the changes are. This has led us just over the last couple of years to be able to have some very different insights, I think, into what's going on, which almost always has been complementing and extending what others have been studying for other approaches. For example, the proteomics that we do from analyzing brain has shown us that there are clear signs of changes in brain metabolism, as well as brain inflammation. Again, Dr. Eldek, Dr. Estes, others, Dr. Wilcock, there's so many great things that this team has been doing at UK, looking at brain inflammation and other things. And this allows us to now extend that work into identifying individual proteins, or what we say are community of proteins, hundreds of proteins that could reflect new biomarkers or new therapeutic targets. And while this starts with postmortem human brain, as I'll show you, we've also spent a lot of time trying to translate this into tools that we can study in living people, so that we can use these for early detection of different processes that are going on in brain, And we can also extend the genetic study. So I mentioned the 40+ genes, which now show risk genes. There are a lot more. That's in many ways the tip of the iceberg. What we can do now is you do these sophisticated analyses where we can identify which proteins are individually mediating the genetic influences on the disease. And many of these proteins we can begin to now associate with other conditions, including symptoms like depression that occur in the disease. So these are just all sort of things that have come up from our group over the last couple of years. To just give you a highlight of one type of example, across these brains, we have seen one group of proteins that we were unaware of previously. Their individual proteins are shown here in the circle, and their identity is not so important individually. But these 100+ proteins all seem to behave together across all these thousands of brains we've looked at. And where studies are available, what we're learning is that many of these proteins reflect the immune activation, the inflammation that occurs as part of the disease process. And this is we've seen this in multiple cohorts of hundreds of individuals from across the country. And this is one study from Seattle, a longitudinal study called the Adult Changes in Thought. And what we learned was that this community of proteins increases quite a bit in Alzheimer's disease brain, shown here, versus control brain. But we've also been studying a group of brains that are from people who were studied annually during life, like many of the participants in the Alzheimer's centers. But then at autopsy, after they passed, it was found that they were cognitively normal, but their brains had Alzheimer's disease pathologies. And we call this asymptomatic AD. And we think this is in part due to this preclinical stage of the disease. So this group of proteins is changing before symptoms begin. And there's a very strong correlation with the diagnosis. And then yet another cohort from one of our colleague Alzheimer's disease centers in Arizona, we see the same group of proteins has a very strong correlation with cognitive ability. And so the higher the amount of these protein levels and these signs of inflammation, the more impaired people were cognitively. So these strong associations then give us clues where the next step has been to actually do studies and basic studies, which I'm not going to go into, where we can manipulate each of these genes in animals like fruit flies and mice, or in cells that we derive from humans, to actually determine individually which of these proteins, if any, may be relevant for new treatment approaches. So this, just to summarize what we're learning from our group and from others, and really the worldwide effort from the increased expansion of knowledge about basic science, is that, while this is a complex disease of genetic, environmental, lifestyle interactions, and now social determinants of health, there are a lot of things that are contributing. And there are a lot of complex pathologies happening in the brain. It's far more than amyloid, which is early on this long trajectory that goes from healthy aging to Alzheimer's disease. Dr. Jicha and others at Kentucky have just done a magnificent job helping us learn about the changes in vascular function and blood flow that are also part of this. And much of the cognitive impairment that occurs in stroke and cerebrovascular disease is very much related. Again, the team has done a great work here at UK on inflammation. Everybody around the world is waking up to the importance of inflammation, how complicated this is. So it can be either damaging or protective. And we've got to learn the nuances to really turn that into important therapeutic approaches, as Linda has really nicely shown us. Brain metabolism, neurofibrillary tangles. And then I add this in Kentucky blue for my friend Pete Nelson, the head of the neuropath for at UK. There are pathologies we didn't even realize that we still have so much more to learn. There's a protein that accumulates called TDP43, which Dr. Nelson has really helped organize globally our understanding of how this pathology also contributes to ultimately the cognitive impairment due to synaptic loss and neuron loss. So this is a complex process, and I take that as a huge opportunity for us. And because it gives us a whole new set of to develop tools to improve our ability to detect the disease as early as possible and to turn that into treatments, hopefully as early as possible. And if we can do that, if we can start treatments, 20 years before symptoms are beginning, we can even have sort of weekly effective treatments, but we can push things out. So if we can get people living to 90, 95 without cognitive impairment, we'll let nature take its course and really hopefully protect the golden years for all of us as we age and the next generation. So let me segue now to briefly talk about some examples here, how we can turn this early detection, turn this knowledge into ways to improve early detection. Common ways that are being developed and now used pretty widely across all the Alzheimer's disease research centers and often in clinical practice are brain imaging and spinal taps to measure spinal fluid. I know those are common at UK as well. But there are advances going on across the scope of understanding this disease. We now very well can identify those core pathologies, amyloid plaques and neurofibrillary tangles with these sophisticated PET scans. We're not huge fans of those at Emory because they're so expensive. And I always try to keep my eye on what's going to disseminate worldwide in an affordable, scalable way. For treatment, and these things are just not available in most places, and nobody's going to be able to afford them. But they're absolutely critical research tools for us because they provide really what autopsy used to provide, sort of gold standard diagnosis. Our center relies heavily on spinal fluid because the spinal fluid bathes the brain and allows us to measure proteins that are floating around in the spinal fluid, which are reflective of these two core pathologies. And as I've just mentioned, our group is very interested in understanding things beyond those two pathologies. I showed you those 100 plus proteins which are present in the brain, even in an asymptomatic stage, that seem to be strongly related to cognition. We started trying to target novel proteins that are also measurable in the spinal fluid. And we took 28 of these proteins that are present in brain and developed ways to measure them in the spinal fluid using mass spectrometry for those who are scientifically inclined. And what we can do, we've seen in early studies that this was going to be a very promising approach, that we can measure proteins and correlate what's in the spinal fluid with what's in the brain, so that these become biomarkers of the actual changes that are occurring in the brain. So over the last few years, we've been spending a lot of time trying to better understand what changes occur in proteins in the spinal fluid. And we've identified really hundreds of proteins which are changed as a result of Alzheimer's disease in the spinal fluid. And because we have a good understanding of the protein changes in brain, we're able to map them. Again, I'm not going to go into details here for the sake of time and understanding who I'm speaking to, but we can now, we sort of reduced a large amount of data so we can begin to think about different proteins that reflect different complex biologies occurring in brain. Some of these proteins reflect changes to brain synapses. Others reflect changes in the cerebrovascular function or blood-brain barrier. Others reflect the white matter. Others reflect this inflammation, and others reflect metabolism. So we're developing these precise protein targets that relate to these different functions, which I showed you are now known to be important in the complex disease pathology. And so we can measure these proteins now and show that they're all in the spinal fluid very, very significantly changed between individuals who don't have Alzheimer's disease and those who do have Alzheimer's disease. And so each of these panels of proteins shows very, very dramatic differences between disease states. One of the biggest challenges we've had in the field is to replicate things, to make sure we can get large groups of people together from across centers and confirm things. And these protein changes in the spinal fluid we've been able to show across 4 other replication studies, different cohorts now, that this is a very reproducible finding. So I'm going to, just for the sake of time, pass on that. One of the, what we wanted to do to be able to scale this is we've adapted a more targeted method, which allows us to measure individual proteins at scale pretty rapidly. And so one of the studies that we're involved in is what's called the autosomal dominant form of Alzheimer's disease, where all of our centers get together and get these rare cases where there are mutations in the amyloid precursor protein or presenilins that cause this young aggressive onset form of Alzheimer's disease. And in these studies, we know there are many young people who are not even symptomatic, but some carry the gene, which we know is going to cause them to develop Alzheimer's disease in the decades ahead. Others in participating in the study are siblings who do not have the gene. So we've been able to study almost 900 spinal fluid samples from these individuals over time and test how these new protein biomarkers in the spinal fluid that we've discovered, how they perform, how they might be able to predict Alzheimer's disease. And we're very encouraged because many of them are showing changes and telling us that the biology, like inflammation or metabolism, are actually getting changed in living people before symptoms begin. So these become new biomarkers as well as potential targets, so we can monitor whether new therapeutic approaches are working or not. The really exciting progress in the field has been, we recognize that spinal fluid is not something everybody wants to give up. We do it routinely, as you guys do at UK, but clearly we would all love to see simple blood tests, which can provide the same benefit. And the field has seen just tremendous progress. So we really are on the precipice of having available in the clinic and research studies simple blood tests that will let us measure both the amyloid plaques and the neurofibrillary tangles, I think, in the years ahead. And the same pipeline that I've shown you for looking at these other biomarkers in spinal fluid, we're now beginning to do in blood as well. And we've had some pretty good initial success. So we're hopeful that we'll be able to, in the future, envision a whole panel of blood tests, which can help us understand the amount of inflammation or metabolic changes or synaptic injury that are occurring together with the amyloid or the neurofibrillary tangles that define the disease. Most importantly, we all want to be where we can actually do more than detect the disease. And the reason we do this is so we can think more intelligently about how to intervene. And it's really important to remember that we have a lot of risk factors that are identified. It's a real challenge for the field to turn risk factors into treatment approaches, but it's a really important area that needs to continue to grow. As I showed you, the epidemiologic studies, there are a lot of risk factors that are identified, and there are a lot of protective factors that are identified. The way I think about these is that they sort of are in a balance and push-pull in terms of determining when somebody is going to get the disease or how rapidly it will progress for them. So these are things that are potentially modifiable. And one of the most important studies, I think, over the last couple of years in clinical trials was something called the SPRINT trial, which had over 8,000 individuals where blood pressure was being assessed. And there's been a lot of questions about how aggressive we should be with treating blood pressure just for medicine. They really designed to protect the heart and protect against stroke. But these 8,000 plus individuals were followed out over years with either the standard care where blood pressure is allowed to, basically be treated to a systolic up to about 140, or more aggressive blood pressure control, where the goal is to get blood pressure systolic under 120. And this was really the first study that I'm aware of that really convincingly showed how important blood pressure control can be. Because what it showed is that there was a reduced risk of progressing to mild cognitive impairment and dementia in normal people as a result of the aggressive blood pressure control. So that's something that can be done today. So I encourage people to be aggressive about getting their blood pressure controlled. And I was really delighted to see Donna, and the team at UK participated, excuse me, in thinking about how these blood biomarkers are going to be a new generation, new opportunity to understand who should be treated and when they should be treated and if the treatments are effective enough. So I think these biomarkers are going to be very complementary to thinking about how lifestyle might modify risk. And so there are a whole host of things other than blood pressure which have been identified as potentially modifiable, including education we've known has a big impact. There are clues that even hearing loss in midlife could be a risk factor for Alzheimer's disease. And in later life, depression, physical inactivity, and social isolation and smoking are all really important risk factors that are all potentially modifiable. So there's a lot of work that needs to be done, but most of these things come with zero risk and a lot of general health benefits. And so I think the more we can encourage people to adopt these healthy lifestyles throughout their life, the better we're going to be. We also know that lifestyle is not going to be the end all. We need to develop better medicines, without a doubt. And this is a figure from a review from one of our colleagues, Dr. Jeff Cummings, who's led a lot of clinical trials in our field, who periodically summarizes all the different treatments ongoing. The only point I want to make from this slide is that we now have over 120 different clinical agents, drugs, therapies being tested at various stages, from early stages to late stages. I think there are at least a couple drugs developed by Dr. Van Eldik that are now in humans as well, that are on this. And so this pipeline has to grow, but I'm very encouraged to see that the numbers of new things being tested continue to increase every year. It's a long slog developing a new drug. I'm sure Dr. Van Eldik will confirm that. It's often said to be a 20 to 25 year journey from the time somebody has an idea to develop a new drug to a new protein target, for example. One of the things that I think our field is underappreciating is the opportunity to repurpose medications. In the cancer field, a lot of things are being tested that have been FDA approved for general medical conditions like blood pressure or diabetes control or antidepressants, et cetera. And in cancer, these things are often being now tested and sometimes being found to be effective for various forms of treatment. These exact same mechanisms provide opportunities for Alzheimer's disease that I think are underappreciated. My colleague at Emory, Dr. Ihab Hajar, is a geriatrician and has been studying cardiovascular risk factors for many years. He has really helped me open my eyes to how important these can be. And so Dr. Hajar looked at our Alzheimer's Center participants from across the country, including at UK, and looked at those who were taking one of these blood pressure medicines. It's a certain class of medication called renin-angiotensin blockers. And so this class of medicines, what you're looking at are people in the national consortium called NAC, who were non-users in blue, and the people that took this one class of blood pressure medicines in red. And what Dr. Hajar saw is that the progression of their dementia, the severity of their dementia rating, was less for those people taking that medicine. He also used one of the other national studies that we all participate in called the ADNI. So these are, some of you may be participating in that, where you get repeated brain imaging, some people get spinal taps. And Ihab looked at this cohort and found that those people that were taking the angiotensin receptor blocker medicine class of drugs actually had less progressive increase in their tau, one of those spinal fluid measures of the neurofibrillary tangles and associated with the amyloid plaques as well. And so this was encouraging sort of association data, and it inspired him to do a clinical trial that we finished at Emory. He took 176 people with hypertension and then looked at, so these are people not necessarily even with the diagnosis of Alzheimer's disease, but mild cognitive impairment due to and hypertension, and treated them with this class of medicine specific drug called Candesartan. And what Ihab found in this phase 2 clinical trial is that the Candesartan actually showed some improvement, which is this reduction in time it takes to do this attention task or this sort of executive function task that we call TRAILS-B. So there is a symptomatic benefit in this clinical trial as a result of treatment with this one class of medicine, Candesartan, versus another blood pressure control. And also he's found some benefit in the white matter, suggesting that there may be some protective effects in the brain as well. And then I know I just have a few more minutes left. I'll just highlight another repurposing effort that we did at Emory based on a drug called Atomoxetine. And so atomoxetine is an FDA-approved drug for attention deficit disorder, and it's been used safely in children and adults for many years. But it targets a very selective pathway that's been of interest over the last couple of years in the field. This is really work that was led. We were inspired by my colleague, Dr. David Weinschenker at Emory, who has done a lot of the basic research understanding this group of brain cells down in the brainstem called the locus coeruleus. And this group of brain cells secretes a neurotransmitter called norepinephrine. And this norepinephrine bathes a lot of the brain. It connects with many parts of the cerebral cortex and the parts of the brain involved in learning and memory. But as Dr. Nelson knows, our neuropathologists have identified that this group of brain cells you can see because it has a pigment in it that allows you to visualize it. And that's what these black spots are deep in the brainstem. But in Alzheimer's disease, this group of brain cells is really severely devastated and very early. And one of the most interesting things that we've been learning as a field is this group of brain cells seems to be the first site in the entire brain that develops some pathology, the neurofibrillary tangles. Even in young adults or even youngsters begin to form some pre-neurofibrillary tangle-like changes in this group. And a whole variety of basic research studies have shown that when this group of brain cells degenerates, there are a lot of things that happen throughout the brain. It activates inflammation, it changes the blood-brain barrier, changes the blood metabolism, all the things that are somewhat similar to what actually happens in Alzheimer's disease. And then in the experimental models, it's also been shown that drugs like atomoxetine, which increase the amount of norepinephrine released by these brain cells, can actually rescue some of the symptoms as a result of degeneration of those brain cells. So as that work progressed, we realized it almost felt unconscionable that we shouldn't be trying something like this, which is already approved by the FDA. So we did a clinical trial where we identified people with mild cognitive impairment. And these were people who had their early memory loss due to early stages of Alzheimer's disease that we confirmed by spinal fluid. And we treated them with either atomoxetine or placebo, and everybody got both. So it's what we call crossover. We were able to measure that the drug got into the brain and had very expected strong effects on norepinephrine metabolism, as we expected. I was quite surprised. We actually saw a reduction in the amount of the spinal fluid measures of neurodegeneration or the tangles, the phospho-tau and tau in this trial. And we also did fMRI to look at brain metabolism and FDG PET, two different ways to look at sort of brain metabolism. And what we found is that there was an effective treatment on brain metabolism as well. And then these biomarkers that I've been showing in the spinal fluid, we found significant beneficial trends with many of these panels that I talked about as well. So this was just accepted for publication. So hopefully we'll get this out there, and we'd like to see this replicated and go to the next level, again, as a way to help accelerate and broaden the pipeline of treatments. And final is a really cool study that we got involved with when a young faculty member moved to the biomedical engineering department at Emory and Georgia Tech a couple of years ago to look at non-invasive treatments, which I think have more promise than I realized. And this is Dr. Annabelle Singer. She came after her postdoc at MIT, where she had discovered that if she could create she could stimulate brain cells at a certain frequency, so that the brain cells would fire 40 times per second, seemed to have a very beneficial effect on memory and learning, and that she could induce this electrically, but more shockingly, she could flash lights to these mice and get the brain to start firing at the same frequency as the lights flickering at 40 hertz. So it really suggested that if you could stimulate the brain non-invasively with lights, or she also then showed sound, you might be able to modulate brain activity. And what was pretty impressive is they did this in these Alzheimer's disease mouse models. These mice accumulate amyloid plaques, shown here in green. And by flickering the lights or sound for these animals, they were able to show there was less amyloid that accumulated. and that the brain cells, the immune activation occurred as a result to help seemingly clear out the amyloid. So it started suggesting a possible mechanism by which flicker might be actually able to change the actual disease pathology, which is, to me, quite remarkable. So this was early work, but we decided that, again, it's very safe. We wanted to test the potential to do this in humans. So Annabelle, who's a biomedical engineer, came to us and we designed a clinical trial. Colleague Dr. Jim Law helped lead the design of this. This is just very simple. We identified 10 participants who wanted to get involved in this to see if this would even be feasible and tolerated. And so what we did is we hooked these people up with the glasses and earphones and had them take them home so they could use this for an hour a day. And we had a couple parallel groups of the participants where they either had them but didn't have the flicker at the right frequency, or they did at various points in the trial. And we found that people loved doing it. They stuck with it throughout the up to 10 months that people have been studied. We did some of these other similar studies. We did fMRI studies where we look at brain activity across the regions by MRI and saw the treatment was actually improving the connection of two of the important brain regions where there's changes in Alzheimer's disease. We did the same proteomics approach. So we looked in the CSF, the spinal fluid. We could measure the proteins in the spinal fluid and classify pretty accurately whether people were during the placebo phase of the treatment or getting the stimulation. And then one of our colleagues, Levi Wood at Georgia Tech, measured a whole panel of immune markers, measures that the immune system was getting activated, and saw a whole variety of changes. So to me, this is very encouraging that even non-invasive approaches have true potential to modulate real biology, real physiology, and potentially even pathology. I think there's going to be a lot of excitement in the future thinking about a whole variety of approaches and not just pharmacologic. So as I conclude, I hope I've been able to show you that Alzheimer's disease is a complex disease. There's a lot more going on in the brain than the amyloid plaques and the neurofibrillary tangles that we've known about. That the scientific advances are just so exciting. I can't tell you how, there's never been a more exciting time to be a scientist and clinician because of all the great work going on all over. We're learning daily new things about the genetic causes and the way the genetic causes are transformed into the disease and how we might capitalize on learning about that to identify new therapies and biomarkers that are going to help us detect the disease early, even pre-symptomatically, without a doubt, in the years ahead, and that our pipeline is going to continue to grow. So I am, for one, I'm very optimistic, and none of this happens without great researchers like those at UK and great participants like all of you. This has got to be a partnership, and it's so important that we get people to help us in this journey because working together, we're going to get to a cure faster than ever. So with that, I just want to acknowledge just a fantastic group of people I get to work with at Emory. I won't go through their name, and the programs like the Accelerating Medicine Partnership that I talked about have colleagues from really across the country. And we also have industry partners that have put in additional money into the program other than the NIH. So with that, just thank you all for attention and giving up some of your Saturday morning, and look forward to participating in the rest of the discussion.